So next speaker is Ben Greenbaum, who also came to us from the Institute for Advanced Studies, is also interested in quantitating the immune response. And we've given Ben 5 minutes and just asked him to speak faster.
Thank you, and thanks everyone, Adam and Dean Charney, for putting this together, Ramon. It's great to have everyone together like this. So our lab's focus, in addition to a substantial interest in virus evolution, is really on understanding the mechanisms by which tumors are recognized by the immune system. So, we kind of are, just to give a sense of what questions we're interested in. So we're interested in this question of to what extent does immune recognition drive tumor evolution, both in the non-therapeutic and in the therapeutic context. The way we think about this is that, recognition emerges as a tumor evolves, so we need to understand how the immune system shapes tumor evolution. Therapy is the target. The mechanisms by which the immune system is co-opted can fundamentally alter a tumor's evolution. This has been shown over the last decade of immunotherapies. And we're continually trying to exploit insights from virus evolution and from computational immunology, which is often trained on larger viral data sets that have more dynamical evolutionary information and more specificity about immune interactions, and bringing those insights over into immune oncology. So I'll talk about 3 vignettes from our work quite quickly. Next, or do I do that? Okay. As Marta was alluding to, we were a part of a study to try to essentially come up with a modeling approach consistent with the hypothesis that in many cases successful response to checkpoint blockade immunotherapy is driven by recognition of tumor neoantigens that are proteins that come from mutations generated throughout the course of a tumor's evolution. And we came up with a consistent model for that hypothesis, based on essentially weighing the fitness costs of likelihood immune recognition across a tumor's subclones, and therefore trying to extrapolate which patients would be more likely to respond or less likely to respond to checkpoint blockade therapy in the limited number of data sets that are currently available on those topics, which is a constant challenge for the field. Additionally, we spend a lot of time trying to think about how to characterize, in this case, the T-cell receptor repertoire associated with productive recognition of a tumor, or ideally the specificity of T-cell receptor repertoire sequences associated with successful recognition of tumor antigens. This is part of a study on a cohort of long-term survivors to pancreatic cancer, where there seemed to be surveillance of the tumor's neoantigens that led to long-term response, and the immune monitoring on the study was done by Miriam and Sasha Njacek. And just showing the different kind of approaches you can take. It's a very dynamical field. New technologies are coming through all the time, and a lot of people are thinking about how to best assay this question. So, in the slide number on the left, you sort of stimulate someone's blood, and you sequence with a potential antigen, and you sequence before and after stimulation. You get T-cell receptors. You look for them in the tumor, and then in the right, you try to understand the transcriptional profiles associated with a productive or non-productive response. Finally, another topic that we work on a lot, that we've been working on with Nina for almost a decade, I think Stephen showed a picture of us together from a very long time ago, is trying to understand the recognition of RNA by the innate immune system, and in the cancer context that comes up in many areas. In particular, something we put a lot of effort into is the hypothesis that innate activation from de-repressed endogenous RNA is associated with innate activation in both positive and negative ways. And we have a sort of a modeling approach that came from virus evolution that we applied to that question. And this is just some slides from a recent, some figures from a recent publication on this topic, showing that there seem to be both, classes of endogenous RNA that can be associated with a productive response to immunotherapy, and at the same time, classes of endogenous RNA that can be associated with immune suppression. So just to wrap up on our future goals, which are probably collective goals on which many people here will hopefully be a part of. We're interested in developing mechanistic models of therapeutic response and immune activations, matching T cell receptors and neoantigens and important tumor antigens more directly, manipulating the expression of endogenous RNA in the way that we described in ways that can be productive towards tumor recognition, and learning a lot from unique patient cohorts in a field that can often be data limited, such as low mutational burden responders to therapy and high mutational burden non-responders. And finally, something that I think we all need to think about together is rationalizing immunogenomics towards questions in cancer immunotherapy, when many traditional quantitative approaches towards assessing tumors might have been developed for a previous era. So thank you.